When you hear the word chemotherapy, a lot of things probably come to mind. But what if I told you that this cornerstone of modern cancer treatment wasn't developed in a lab to save lives? It was actually discovered on a battlefield as a weapon of war. And the really wild thing is, its success hinges on a fatal flaw that's baked right into the very DNA of cancer itself. So here's our game plan. We're going to start with chemotherapy's absolutely shocking origin as a chemical weapon. Then we'll zoom right down to the cellular level to see how it works and why that attack causes such, well, infamous side effects. We'll uncover the critical difference that lets it kill cancer while sparing the patient. And finally, we'll look beyond the drugs we have now to the future of truly precision medicine. All right, let's dive right in at the very beginning. The birth of one of modern medicine's most powerful tools is, well, it's completely backwards. It starts not with a mission to heal, but with a weapon designed only to cause harm. Yeah, the story of chemotherapy actually begins in the trenches of World War I. And the main character here wasn't a doctor or a scientist. It was a chemical agent that was never, ever intended to save a single life: mustard gas. Right in the middle of all that horror, battlefield physicians started to notice something, a really grim but crucial observation. They saw this bizarre pattern in the blood work of soldiers exposed to the gas, a pattern that would spark a revolutionary idea and completely change medicine forever. Okay, so this timeline really lays out how it all happened. Doctors in the field saw that mustard gas just absolutely wrecked a soldier's white blood cells. This led to a brilliant out-of-the-box question. What else is defined by cells that divide like crazy? Cancer. So in 1942, researchers at Yale went for it. They gave a modified, less toxic version of the compound to a patient with lymphoma. And the result was incredible. The tumor actually shrank. Then, just a year later, a tragic shipping accident in Bari Harbor, Italy, exposed hundreds of people to mustard gas. It was awful, but it provided this flood of undeniable data that confirmed just how potent this stuff was. And that really solidified the foundation for everything we now call chemotherapy. So from this totally unlikely origin, a whole new strategy for fighting cancer was born. See, unlike surgery or radiation, which are really localized, get-the-spot treatments, chemo was designed to be a systemic attack, a war waged on a cellular level, hunting down cancer wherever it might be hiding in the body. So here's the key idea. Chemo drugs travel through your bloodstream to pretty much every corner of your body. Their one and only mission is to find cells that are actively dividing and to stop them in their tracks. For a cell, not being able to finish division is a catastrophic failure. It triggers a built-in self-destruct sequence called apoptosis, basically programmed cell death. And they do this in a few different, pretty clever ways. The first class, alkylating agents, these are the direct descendants of mustard gas. They act like cellular saboteurs. They get in there and directly shred a cell's DNA blueprint so badly that the cell has no way of making a clean copy to divide. Then you have the antimetabolites. These guys are the imposters. They're designed to look exactly like the essential nutrients a cancer cell needs to grow and divide. The cancer cell, being greedy, gobbles up what it thinks is food, but it's actually poison, which grinds the whole replication process to a halt. And then there's a third strategy, which is a bit more mechanical. Mitotic inhibitors, they don't mess with the DNA or the food supply. Instead, they sabotage the physical machinery of division itself. You can think of it like throwing a wrench into the gears of a factory just as it's about to finish a product. It prevents that one cell from completing the critical act of pulling itself apart into two new cells. Now, this strategy of attacking any and all rapidly dividing cells is incredibly powerful, but it comes at a huge cost. It leads directly to the treatment's harsh reality and to the one question that pretty much everyone asks when they think of chemo. And the answer, in 2 words, is collateral damage. Chemotherapy is a blunt instrument. It's fantastic at killing cells that divide quickly, but here's the problem. It cannot tell the difference between a malicious cancer cell and a perfectly healthy, normal cell that just happens to divide quickly as part of its day job. And this table really breaks it down for you. That crushing fatigue? That's chemo hitting your bone marrow, slowing down the production of your oxygen-carrying red blood cells. Hair loss? Well, it happens because hair follicles are some of the fastest-growing cells in the entire body. The nausea comes from irritation to the stomach lining, another area with high cell turnover, and from the drugs literally triggering the toxin detector in your brain. Every single major side effect is a direct result of chemo damaging healthy, hard-working tissues. And we're even starting to understand some of the weirder neurological effects, like chemo brain or that pins and needles feeling in the hands and feet. It seems to be caused by a different kind of damage. Here, the drugs are harming the mitochondria, the tiny power plants inside our nerve and brain cells, basically causing an energy failure that leads to all these frustrating symptoms. So, okay, if chemotherapy is hurting both healthy cells and cancer cells, why isn't it just a death sentence for both? Why does it work at all? Well, the answer lies in one critical, fundamental difference between the two. And this, right here, is the secret sauce. This is why it all works. Chemotherapy brilliantly exploits cancer's single greatest weakness, its own genetic chaos. You see, your healthy cells have all these quality control systems, these biological repair kits. When chemo damages their DNA, they can hit pause, fix the problem, and then recover. But cancer cells, by their very nature, have broken repair kits. When chemo shatters their DNA, they have no ability to stop and fix it. They just foolishly try to divide anyway with all this damaged genetic material, triggering what scientists call a mitotic catastrophe. The cell literally rips itself apart from the inside. It's brutal, and it's beautiful. And that incredible ability of your healthy cells to recover, that's the whole reason treatment is given in cycles. That rest period between doses isn't for nothing. It is a strategically vital window. It's when your healthy tissues, following this 2-month rule of thumb, get to repair and regenerate, getting stronger for the next round, all while the damaged cancer cells just continue to die off. But a successful treatment isn't just about the drug itself. A huge part of the whole equation is managing this delicate balancing act inside the patient's body and understanding all the other factors that can dramatically change the outcome. Before a single drop of chemo is ever given, there's a whole series of critical safety checks. A blood count makes sure the bone marrow is strong enough to take the hit. A metabolic panel confirms the liver and kidneys can actually process these powerful drugs. Even a dental check is vital, because you don't want a tiny infection becoming life-threatening when the immune system is down. It's all about making sure the patient is ready for the fight ahead. And you've got to remember, it's not just about the drug. Where a cancer starts can be just as important. Think of it like real estate: location, location, location. A tumor in a vital, hard-to-reach organ like the pancreas is immediately way more dangerous. Cancers that start in the lungs or liver, the body's superhighways, have a much easier path to spread. The organ of origin really dictates everything, from surgical options to how fast the disease might progress. So all of this brings us to right now and to the future. For decades, chemotherapy has been that effective but very blunt instrument. But the future of oncology is moving decisively away from that carpet-bombing approach and towards something much more like a guided missile. You see, the whole question that oncologists are asking is fundamentally changing. Traditional chemo asks every single cell, are you dividing fast? If the answer is yes, it attacks. Targeted therapy asks a much, much smarter question. It asks, do you have this very specific mutated protein that only a cancer cell would have? If the answer is yes, it attacks. If no, it leaves the cell completely alone. It's the difference between carpet bombing a city and a laser-guided strike on a single building. So how do these guided missiles actually work? Well, it's pretty amazing. These therapies, called antibody-drug conjugates, or ADCs, are essentially biological smart bombs. First, an antibody is programmed to seek out one specific protein that's only found on the outside of a cancer cell. Then, a super potent chemo drug is attached to that antibody. The whole package circulates harmlessly through the body until the antibody finds its target, latches on, and gets pulled inside the cancer cell, delivering its toxic payload directly and killing it from within, all while leaving healthy neighbors totally untouched. This incredible shift, from a systemic poison to a personalized weapon, leaves us with one last really big thought. For 100 years, we've defined cancer by its location, lung cancer, breast cancer. But as we get better and better at treating it based on its specific genetic signature, will we stop thinking about it by where it is and start thinking about it by what it is? The future might not just change how we treat cancer, but how we even define it.